Hi, everyone. The video you are about to watch is part of the Age Presents lecture series. The series is produced by the American Aging Association to provide an introduction to the foundational and advanced topics in geroscience. As you watch this video, I would like for you to keep 3 points in mind that will help you understand the importance of biological aging. 1, aging is the primary risk factor for disease, including cancer, metabolic syndrome, and most neurodegenerative diseases. 2, the biological approaches that you will learn here today, such as model systems that help researchers learn more and understand about these types of diseases. And 3, mostly deals around you. What is your interest in aging research? Whether it's biological, translational, or clinical, how would you like to contribute to the field? And if you're not already a member of the American Aging Association, I would encourage you to visit our website to learn more about us and consider joining today. AGE members receive free access to the Society's journal Geroscience, reduced registration for our annual meeting, opportunities to interact with many of the leading scientists within the field, and access to these lectures, slides, and notes used here today. And there's more. We have a very active trainee chapter with many opportunities for networking and career development. And last but not least, I would like to thank our sponsors for the Age Presents lecture series. The University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Aging Center, and the Nathan Shop Centers of Excellence at both of these institutions. Thanks for listening and enjoy the lecture.
Hi, I'm Ashley Webb, and welcome to the Age Presents lecture on stem cell exhaustion. So this is an outline for the lecture today. So I'll begin with an introduction to stem cells, followed by a brief discussion of the stem cell theory of aging. And then I'll spend the bulk of the lecture on the mechanisms of stem cell aging and what we've learned about these mechanisms and their impact on accelerated aging of stem cells. And then I'll wrap up with a summary of where the field is in terms of the potential for rejuvenating stem cells to improve healthy aging. All right, so stem cells have 2 basic properties. They are able to self-renew and they're able to terminally differentiate. So for example, this stem cell here depicted in this schematic is able to divide and regenerate copies of itself through this process of self-renewal. It also can go on to generate cells that will terminally differentiate. And before they do so, in general, stem cells will go through a proliferating precursor phase shown here. And this precursor phase is important because it actually allows for the amplification of the stem cell pool so that a small number of stem cells can actually generate a large number of terminally differentiated cells in a tissue. And so terminally differentiated cells in a tissue are basically at the end of their developmental path. So these terminally differentiated cells have specialized functions depending on the tissue in which they reside. So for example, neural stem cells will generate neurons in the brain that are terminally differentiated and post-mitotic. So generally speaking, although there are exceptions to this rule, terminally differentiated cells are post-mitotic and they do not re-enter the cell cycle. So I'd like to just mention a few different features of these adult stem cells. So adult stem cells are also known as tissue-specific stem cells or somatic stem cells. And so this distinguishes them from embryonic stem cells, which are present early on in development and can give rise to all of the different lineages in the embryo. But all stem cells have the ability to self-renew, as I mentioned, and they do have the capacity to differentiate into specialized cell types. Now, adult stem cells are relatively rare in tissues, but as I mentioned, they will go through a proliferating phase where they can generate more precursor cells, which allows a small number of these rare stem cells to give rise to lots of differentiated cells as needed in the tissue. And so these rare stem cells reside in specialized niches, and later in the lecture, I'll give some details on what's happening in stem cell niches. And for the most part in the adult, stem cells are maintained in a quiescent state. And so quiescent stem cells are in a state of relative dormancy, and they have to first be activated and enter the cell cycle in order to go on to differentiate and contribute to tissue repair or regeneration. But these actively proliferating stem cells basically have a decision to make. They can either go on to differentiate into terminally differentiated cells, or they can renew the pool of quiescent stem cells. And so it turns out that if you look across different tissues are actually renewed at different rates. So basically within any given tissue, there are specific needs or demands of the stem cell pool that are particular to that niche and environment. So some tissues in mammals are tissues that have constant turnover or high turnover. So examples of this are the hematopoietic system, where the stem cells, which are hematopoietic stem cells, reside in bone marrow, as well as intestine. Intestine is a very high turnover tissue, and the stem cells reside in the crypt of the intestine, and they're very important for regenerating the epithelial of the intestine. The skin is also a high turnover tissue, and stem cells are critical for maintaining healthy skin during the aging process. Some tissues have much lower turnover. So the brain is an example of this. Though there are two specialized neural stem cell niches, and neurogenesis, so the formation of new neurons from stem cells in these niches, is important for healthy aging and maintenance of cognitive function and sensory functions with age. Now in the skeletal muscle, the stem cells are known as satellite cells, and these cells are important for the repair process after damage to skeletal muscle. Now, stem cell exhaustion is one of the major hallmarks of aging. So most people are familiar at this point with this concept of hallmarks of aging. So these are processes that are involved in aging and decline in tissue function with age. And so stem cell exhaustion meets the criteria as a hallmark of aging. And so that means that it is observed in normal physiological aging, and that defects in stem cell function can actually accelerate tissue aging, and that rejuvenation of stem cells, or if you increase their functionality, that can actually enhance repair or improve aging symptoms within that tissue. And so the stem cell theory of aging is one of several theories of aging, and it's just one contributor to the aging process. And so the stem cell theory of aging posits that aging is a result of the failure of stem cells to replenish cells across tissues, and that this results in a loss of tissue and organ function during aging. And so, as I mentioned, aging is accompanied by stem cell defects that affect various tissues across the body. And stem cell proliferation generally decreases with age. However, there are exceptions to this rule, and I'll mention some exceptions in upcoming slides. Now, during aging, there's also a skewing of stem cell lineages. And so, as I'll mention in upcoming slides, some stem cells will actually generate different types of differentiated cells with age. It's also now known that there's an overall depletion of stem cell pools with age, and that this is due to a loss of quiescent cells. And so once these quiescent stem cells are lost, this is very detrimental to the lineage, because there's no longer a reservoir for differentiated cells once these quiescent stem cells are depleted. It's also been shown that the quiescent stem cells that remain in the aged body will actually drift deeper into a quiescent state. And so this is also detrimental to the lineage, because it means that these quiescent stem cells are actually much harder to activate. So it's harder to get them out of the quiescent state into the actively proliferating state in order to go on and differentiate and contribute to repair. So all of this together results in a failed maintenance of tissues, as well as a decrease in regeneration of tissues with age. So a lot of what we know about the stem cell theory of aging and support for stem cell theory of aging comes from normal physiological aging and observations in terms of what happens to just normal stem cells during the normal aging process. However, there's also evidence for this theory from accelerated aging syndromes and accelerated aging models. So for example, Hutchinson-Gilford progeria is an accelerated aging syndrome that results from mutations in the lamin A gene. And so this individual here has Hutchinson-Gilford progeria, and you can see he exhibits a number of features of accelerated aging, although he's quite young. So for example, there's a thinning and wrinkling of the skin that occurs, there's decreased muscle mass, increased joint stiffness, as well as an overall decrease in lifespan. And so there are also mouse models of Hutchinson-Gilford progeria. An investigation of these mouse models has confirmed that there's a depletion of stem cell pools in this progeria. So for example, here, looking at a section through a hair follicle, in wild-type mice, there are a large number of stem cells that are present in the hair follicle, and those are shown here in red. But in the progeria model that recapitulates the symptoms of lamin A mutations, there is a depletion of these stem cell pools. So there's a loss of the red signal here. And this isn't limited to just Hutchinson-Gilford progeria. It's also been observed in Werner syndrome, another progeria. And even in the individuals that have these syndromes, there's a depletion of mesenchymal stem cells that have features of premature aging. Okay, so what are the tissue-specific features of stem cell aging? So, so in hematopoietic stem cells, for example, there's an overall increase in their number with age. And in addition to that, there is a myeloid bias. So this is that lineage skewing that I mentioned. And the increase in HSC number, together with the myeloid bias, is what increases the presence of myeloid dysplasias with age in humans, as well as the increase in leukemias, myeloid leukemias with age. And so as a consequence, in aged individuals, there are fewer adaptive immune cells. And there's also evidence for increased immunosenescence, as well as a clonal skewing in the hematopoietic system. Now, this is in contrast to other tissues or compartments, such as the brain and the germline, where there's an overall depletion of the stem cell pool that's been observed. Now, in the brain, for example, in addition to this overall depletion of the stem cell pool, there's a reduction in new neuron formation and a bias towards glial cells. So with age, neural stem cells actually make more astrocytes than they do neurons. So that's a lineage skewing that occurs. And so this depletion of the stem cell pool, as well as the reduced neurogenesis, correlates with decreased cognitive function. And other stem cell compartments are aging yet differently. So for example, skeletal muscles and satellite cells within skeletal muscle have been observed to acquire deficits during aging. And the result of this is an impaired regenerative process, in particular in response to damage, as well as a skewed differentiation potential. Now, the skin is also well known to exhibit features of aging, and there are defects in stem cells that are responsible for these age-associated deficits. So for example, there are defects in wound healing, and as you know, there's a thinning and wrinkling of the skin, as well as an overall hair loss with age. Now in the intestine, there's also strong evidence for impairments of stem cell function. A lot of what we know about intestinal stem cells actually comes from work in Drosophila and invertebrate systems. And it's a little bit less clear exactly what's happening to intestinal stem cells in mammals with age, but it is well documented that in general, there is a functional decline of the intestinal stem cells. And this results in a loss of epithelial homeostasis, as well as an increase in inflammatory diseases and dysplasias. Okay, so I'll spend the next few minutes discussing the mechanisms of stem cell aging. And so the mechanisms of stem cell aging can be broadly grouped into intrinsic mechanisms and extrinsic mechanisms. So the intrinsic mechanisms of stem cell aging include changes in mitochondrial function and metabolism, as well as increased DNA damage and telomere attrition. There's also evidence for defective proteostasis and turnover of damaged organelles in stem cell aging, as well as epigenetic changes that are referred to as epigenetic drift that are accompanied also by changes in chromatin architecture. It's also known that, that during aging, there's an increase in cellular senescence in stem cells themselves, and in some cases, an increase in apoptosis. An emerging area in the stem cell field is in the investigation of how circadian regulation and circadian rhythms are altered during stem cell aging, and how this may reduce the functionality of stem cells with age. And then finally, nutrient sensing and transcription programs are well known to be altered in stem cells with age and contribute to the decline in stem cell functionality. All right, so mitochondrial dysfunction and alterations in metabolism have been well studied across tissues in the context of aging. And stem cells have problems with their metabolism with age as well. And so it's important to note that stem cells are actually highly metabolically specialized cells. And even comparing quiescent stem cells to active stem cells or actively dividing stem cells, there are many differences, metabolic differences that are apparent. So for example, quiescent stem cells are more biased towards glycolysis, whereas actively proliferating stem cells have higher oxidative phosphorylation. And this is very important because it helps to meet the needs of these actively proliferating cells, which have higher metabolic needs than the cells that are in a more dormant state. Lipid metabolism has also observed to be different between actively dividing stem cells and quiescent stem cells. And this has been shown in neural stem cell pool, where lipid metabolism is critical supporting the dividing state. So it'll be interesting to see across other tissues if that's a conserved feature. So like I said, similar to other tissues and cell types, mitochondrial damage and altered metabolism is a major feature that accumulates with age. And so there's an increase in reactive oxygen production that occurs with stem cells, and this causes mutations in mitochondrial DNA. And there is evidence that these mutations in mitochondrial DNA actually cause stem cell dysfunction, because mutator mice that have high levels of mitochondrial DNA damage actually have defects in their hematopoietic stem cell and neural stem cell lineages. And this correlates with premature aging. It's also been shown that there are alterations in the production of metabolic cofactors during the aging process. And this is important because many metabolic cofactors have been coupled to gene expression. So for example, acetylases and methylases, which are critical for depositing epigenomic marks across the genome on DNA or histones, require these metabolic cofactors. In addition, these metabolic cofactors are critical for post-translational modifications that maintain protein function during healthy aging. And there's actually evidence that, that replenishing pools of metabolic cofactors can actually assist in healthy aging and improve the aging process. So for example, treating mice with nicotinamide riboside, which is a precursor to NAD+, can actually rejuvenate the muscle stem cell pool in aged mice. So other features of mitochondria and metabolism that are altered with age include just an overall disruption in energetics, as well as an activation of the unfolded protein response that's been observed. All right, so altered proteostasis and increase in organelle damage is another major feature of stem cell aging. So proteostasis, which is also known as protein homeostasis, is the process by which cells are able to maintain the proteome, so the abundance and the stability of proteins in the cell. And it's well known that cellular stress increases with age and is known to also cause protein unfolding or misfolding. And so, unfortunately, proteostasis is impaired with aging, which allows for increased damage and aggregate formation. So this includes both the accumulation of damage in organelles, as well as aggregated proteins that are associated with normal aging, as well as neurodegenerative diseases. And so this schematic of a cell here just depicts the distinct systems of proteostasis in the cell. So for example, autophagy is sort of a recycling center for the cell. And so this is a way for the cell to collect damaged organelles or damaged or aggregated proteins and actually deliver them or accumulate them into autophagosomes that fuse to lysosomes and then eventually degrade these damaged components. And then the biomolecules can actually be reused by the cell. So it's sort of a recycling system in that way. So molecular chaperones are also present in the cell, and these are important for handling unfolded proteins as well as misfolded proteins. And in addition, the ubiquitin proteasome system is critical for getting rid of damaged proteins through tagging with ubiquitin and then delivery to the proteasome for degradation. So there's quite a bit of evidence now that these proteostasis systems are important to maintain healthy aging of stem cells. And interestingly, it turns out that quiescent and activated stem cells actually have different protein quality control systems in order to stay healthy. And so this was shown, for example, by Andrew Ney's lab, where they compared quiescent neural stem cells and activated neural stem cells. And just looking at staining of lysosomes and accumulation of aggregates in the lysosomes, it was shown that there's a large accumulation of these huge lysosomes in the quiescent neural stem cells that actually sort of hold on to and harbor these aggregates during the aging process. And so they're basically protecting the cells from damage by collecting the aggregates in the lysosomes. Now, the activated neural stem cells, in contrast, actually turn over these aggregates rapidly. So they're actually actively cleaning the cells instead of just protecting the cell by collecting them. And so, so these large aggregates, or these large lysosomes containing aggregates, are no longer present in these activated neural stem cells. And so during the aging process, impaired autophagy has been linked to stem cell defects. So for example, mice that have that lack autophagy systems due to a knockout of either ATG12 or ATG7 have been shown to have deficits in hematopoietic stem cell aging, as well as satellite stem cells during aging. And this has been shown to be due to an accumulation of damaged organelles, such as damaged mitochondria, in these mice, resulting in increased cellular senescence. So in general, maintenance of healthy proteostasis is critical for healthy stem cell aging, and other proteostasis processes have also been linked to stem cell aging. So for example, the unfolding protein response in the ER is known to be important for maintaining stem cell health with age, as well as segregation of damaged components. So damaged proteins and organelles such as mitochondria can be asymmetrically segregated to reduce damage in stem cells and maintain a healthy stem cell lineage with age. Right, so another feature of aging stem cells is the presence of epigenetic changes, including epigenetic drift. And so it's been observed across multiple stem cell types that there are alterations in DNA methylation that occur with age. And in addition, it's been observed that there's an increase in repressive covalent histone modifications across nucleosomes. And that's been found in both muscle satellite cells as well as hematopoietic stem cells. And so there are consequences of these epigenetic changes for the stem cells. So, so these changes have been associated with transcriptional noise, as well as a skewing overall of expression programs. And so loss of tight regulation of transcriptional programs results in impaired stem cell function with age. And in some cases, these alterations in transcriptional programs can actually result in a clonal dominance, such as in the hematopoietic system. So the idea here is that in some stem cells, epigenetic drift or small epigenetic changes can actually confer proliferative advantages to small populations of cells that then become amplified and eventually dominant as a clone over time. And so these changes in epigenetic states and the epigenetic drift are known to be caused by metabolic changes. So for example, I mentioned there are changes in mitochondrial and metabolic cofactors that are well known to be linked to enzymes that are critical for depositing epigenetic marks across the genome. In addition, epigenetic drift has been linked to somatic mutations. And so as a result, it's been observed that there is an increase in inflammation, an increase in clonal dominance in stem cells with age and in stem cell lineages with age. And it's also been linked to increased cancers such as leukemias and skin cancers. And so this is just a schematic depicting this concept of epigenetic drift. So in young stem cells, epigenetic states remain tight, whereas as these stem cells age, there's a slow and gradual epigenetic drift that occurs. And if this epigenetic drift becomes persistent, then it can result in the deregulation of the epigenome, resulting in transcriptional noise and slow alterations in the transcriptional programs that cause tissue dysfunction with age or stem cell dysfunction with age. Now, in addition, these old stem cell populations can actually acquire somatic mutations. And if those somatic mutations happen to be in genes that encode for important epigenetic modifiers, such as DNMT3A, TET2, and so on, changes in the activity of these chromatin modifiers can further impair the epigenetic program in the cell. And this can result in not just tissue dysfunction with age, but also transformation and cancer. So a really exciting and emerging area in the stem cell field is in the study of circadian control of stem cell function. And so the circadian clock is, interestingly, actually mostly maintained in a cell autonomous manner. However, circadian clocks are responsive to environmental cues. So for example, clocks can be shifted by light, and they can also be shifted by behavioral changes, such as changes in feeding patterns, sleep timing, or activity changes. And this is important because the circadian clock is important for the regulation of cell division. And so the circadian clock has been implicated in the maintenance of healthy stem cells, including in the maintenance of healthy adult neurogenesis. So one of the proteins that's critical for maintaining oscillations in the circadian clock is known as period. And it turns out if you look just at the neurogenic niche over time across a 24-hour cycle, the activity of period, by looking at a reporter of period activity, the activity is actually oscillating across a 24-hour period, where activity of this reporter is higher during the day and lower at night. And these oscillations in gene expression and period activity actually correlate with changes in neural stem cell activity, so neurogenesis and cell division. And that's shown down here. And so wild type mice, looking at in the dentate gyrus, which is a neurogenic region of the adult brain, wild type mice actually have low stem cell division during the day, corresponding to high period expression, and then higher stem cell activity at night when period activity is low. And interestingly, mice that have period ablation, so knockout mice for this period gene, have an overall increase of stem cell activity during the day, as well as a slight flattening of these oscillations. Ablation of other genes that are also known to impact the circadian clock, such as BMAL1, also result in defects and deregulated in stem cell function and activity. Okay, so what happens during aging? So during the aging process, there's an overall functional decline of a region of the brain known as the suprachiasmatic nucleus. And this is a region of the brain that's in the hypothalamus, and it's the important region of the brain that functions as sort of a master clock for circadian rhythms. And in most stem cell compartments, there's no evidence, or nobody's really looked at whether there's a circadian dysfunction or circadian regulation during aging. But it turns out that during aging, where people have looked, circadian oscillations are maintained in a cell autonomous manner, but they're less responsive to changes. And it's likely that these circadian oscillations are less responsive to changes due to the functional decline of the suprachiasmatic nucleus. Now, I mentioned that that in most compartments, we just don't know yet if there are changes in stem cell function due to changes in circadian rhythms. But we do know that in aged epidermal and muscle stem cells, there's actually a relative maintenance of core circadian machinery. But overall, the downstream transcriptional network that's regulated by these oscillating circadian proteins is rewired, and it's rewired in a way that prepares cells for better stress adaptation, suggesting that there are actually changes in circadian clocks with age, and this impacts stem cell function. And so the details of this schematic aren't important, but it's just to demonstrate that there's a transcriptional circuitry that includes this period protein as well as the BMAL1 protein that's critical for maintaining these oscillatory gene expression networks with age. And then there's a rewiring of these networks that occurs to allow cells to adapt better to stress during the aging process. All right, so DNA damage and telomere attrition are also features of stem cell aging. And this was shown a number of years ago by Derek Rossi, where he observed that there's an increase in gamma H2AX foci that occur in hematopoietic stem cells with age. So that's shown here in green. So the green represents the gamma 2AX staining, and the blue is the DNA, so that's DAPI staining. And the result is quite striking if you look at the quantification of these results on the right, where the gray bars represent the aged animals and the overall number of gamma H2AX foci. And as you can see, relative to young animals, which have almost no damaged foci, the aged HSCs actually accumulate quite a number. And so there are a number of different causes of this increased damage with age. So for example, there's an increase in replication stress, particularly in the activated pool of stem cells. And as I mentioned earlier, there's an increase in reactive oxygen species. It's also known that there's telomere dysfunction that occurs with age, as well as genotoxic stress that increases aging features of stem cells. Right, and so as a consequence, stem cells have altered function with age due to these changes in DNA damage and increased DNA damage. So it's been linked to changes in gene expression that overall result in a reduced self-renewal of stem cells, as well as differentiation capacity of stem cells. It's also known that these DNA damage events can lead to arrest of the stem cells, so cycling arrest, and cause them to enter either a senescent state or undergo apoptosis, which causes a loss of the lineage overall. There's also evidence that these DNA damage events can increase oncogenic transformation, particularly in the hematopoietic lineage. However, the good news is that in some cases, stem cells are able to repair the DNA damage that accumulates with age. And this is possible because as DNA damage accumulates, checkpoints are activated within the cell that slow the cell cycle and allow for repair of the damage. And interestingly, depending on which type of cell type or which type of stem cell you look at, there are different repair pathways that are involved in addressing this damage. So for example, in actively cycling stem cells, such as intestinal stem cells, these cells tend to use homologous recombination to repair DNA damage. Now, in contrast, stem cells that are in a quiescent state, such as hematopoietic stem cells, tend to use air-prone, non-homologous end joining for repair. And that's because the homologous recombination pathway is not available to these cells in a quiescent state. All right, so all of the processes that I mentioned so far that impact stem cell aging, these are all under the control of signaling pathways, as well as transcription factors that have been also linked to healthy aging. And so many of these transcription factors or signaling pathways actually function as nutrient sensors within the cell. So examples of this are mTOR and AKT, which have been linked to stem cell function. In addition, transcriptional regulators, such as the FOXO transcription factors, which have been linked to aging across species and across tissues, are known to be important regulators to maintain healthy transcriptional networks during the aging process. So for example, FOXOs are well known to control gene regulation of genes involved in proteostasis, such as autophagy and proteasome activity, as well as resistance to stress, such as oxidative stress. And so a number of studies investigating mouse models, as well as human systems, have linked changes in the levels or activity of these various factors to both alterations of these downstream activities, as well as changes in stem cell function during aging. Okay, and I just wanted to mention briefly that, that quiescent stem cells actually have specialized mechanisms that help reduce damage during aging. And this is important because the quiescent stem cells are the reservoir of stem cells that provide eventually the differentiated cells within the tissue. And so if these quiescent stem cells accumulate damage, that damage is going to be carried through to the differentiated state and impair the repair process. So quiescent stem cells are known to have reduced DNA damage because they're not in a proliferating state. They also have lower metabolic activity compared to actively proliferating stem cells, and they have low levels of transcription and translation. It's also been shown, as I showed earlier with the quiescent neural stem cells, that, that they're able to sequester aggregates in lysosome to help protect the quiescent cell. And they have specialized transcriptional programs that help to maintain the quiescent state and keep these cells healthy and relatively clear of damage. Okay, so all of these intrinsic mechanisms that I've summarized so far are actually impacted by extrinsic mechanisms and extrinsic changes that can occur with age. And so these include systemic changes across the body and in the circulation, as well as local changes within the stem cell niche and changes in the environment. Okay, so local changes occur due to changes in the microenvironment of the stem cell. So this is really the stem cell niche where stem cells reside. And on the left here, this is a schematic, a sort of a generalized stem cell niche. And the stem cell is shown here in pink. And so stem cells are in contact with various cell types around them, depending on the particular stem cell niche. And these cells around the stem cells, surrounding the stem cells, are critical for the delivery of molecules instructing the stem cells to either stay put or to go on to enter the activated state. Once the stem cells enter the activated state, there's actually feedback from the progenitor cells also to maintain healthy levels of the lineage and to generate new cells within the tissue only as needed. Another major feature of the stem cell niche is the presence of extracellular matrix. So of course, cells are embedded within an extracellular matrix, and changes in the extracellular matrix have also been linked to changes in stem cell function. The stem cell niche across multiple tissues has been shown to also include blood vessels running through the niche. And I'll mention the importance of these blood vessels in the next slide when I discuss the soluble factors and circulating factors that are known to impact stem cell function with age. And so in general, the stem cell niche is a really critical and microenvironment for these stem cells. And it's known to help with structural support, trophic support, as well as topographical information, and to provide physiological cues to maintain the stem cell lineage. So during aging, there are a number of changes to the stem cell niche that affect stem cell function. So in some cases, there's an altered composition of the stem cell niche. There's also an increase in overall tissue inflammation. So for example, in the brain, there can be an increased activation of microglia that impact stem cell function. The increase in the presence of senescent cells in the niche is also known to impair stem cell function through secretion of senescence-associated secretory proteins, or the SAS. And as I mentioned, extracellular matrix changes are also a feature of the aging stem cell niche. So for example, in the muscle, there's a loss of fibronectin that's been observed. So altogether, this results in altered mechanical properties, signaling factors, and growth factor availability in the niche that impairs stem cell function. And this is just one example of a schematized niche in the brain and some of the changes that occur during the aging process. And so this is the subventricular zone niche of the adult mouse brain. And while in the young adult, there are abundant stem cells shown in purple here, there's a reduction in the overall number of stem cells with age, as well as an overall thinning of the niche. And like I said, changes in composition can occur. So that's true in the ependymal layer of the subventricular zone niche in the aging mouse brain. Okay, so what are the systemic contributions to stem cell aging? So circulating factors have been shown to have a major function in maintaining a healthy stem cell niche and contributions to stem cell decline during aging. And so systemic changes include changes in hormones, nutrients, as well as increased pro-inflammatory factors, and even changes in the microbiome that can impact stem cell aging. And so this work began a number of years ago from an important study from Tom Rando's lab by Irina Convoy, where it was already known that with age, there are impairments in the regenerative process in muscles. So just looking at a damaged aged muscle, you can see that there's been a failure of repair in this adult aged mouse. compared to a young mouse over here. And it turns out that if you take aged mice and provide them with circulatory factors through heterochronic parabiosis from young animals, you can actually completely rejuvenate repair in the aged muscle. And so that's what's shown here on the in the image on the right. So aged animals that are actually receiving a circulatory system from young animals through parabiosis have rejuvenation of the niche. And so the regeneration index is shown here on the right, and you can see the aged animals are now repairing their muscle in response to damage, similar to what the young animals are able to do. And so this feature of rejuvenation of the stem cell niche from young circulating factors is not restricted to just the muscle stem cell compartment. It's also been shown in the brain, for example, as well as in other areas. So in the brain, Salvieta and Tony Weiss-Correa showed that heterochronic parabiosis also affects levels of neurogenesis and cognitive function in the dentate gyrus. And so in the upper left here, you can see that there's quite abundant neurogenesis in the young brain, and with age, there's a decrease in the number of new neurons that are forming. So this is based on double cortin staining. And heterochronic parabiosis, where the old animals have received circulation from the young animals, there's actually an increase in neurogenesis. And if you look at the graph on the lower right, you can see that there's about a tripling of neurogenesis in the aged animals. And it turns out that although the neurogenesis is not quite at the level of the young animals, this is sufficient to increase cognitive performance in these animals, suggesting that even a small boost in neurogenesis is able to improve learning and memory functions. And conversely, they also found that the aged circulation was detrimental to the young stem cell niche. It actually reduced neurogenesis in the young animals. And so that's shown in the graph on the left here, where the younger animals undergoing isochronic parabiosis had high levels of neurogenesis, but just introducing an aged circulatory system was sufficient to reduce numbers of new neurons that are forming, as well as performance in cognitive tasks. So this is very exciting because it does suggest that, that the aged stem cell niche can be rejuvenated. And this has been a huge area of interest recently in the stem cell field. And one area of interest in particular is in using iPS-based approaches to rejuvenate stem cell pools. So induced pluripotent stem cells, or iPS cells, are cells that are generated from adult somatic cells or embryonic somatic cells, such as fibroblasts. And so these somatic cells can effectively be reprogrammed through delivery of a number of transcription factors. And this was first shown by Shinya Yamanaka. And so, delivery of these transcription factors causes an overall reprogramming of these somatic stem cells into an embryonic state known as an induced pluripotent stem cell state. So these cells are very similar to embryonic stem cells. And so that means that they actually have the capacity to differentiate into all lineages and all cell types in the organism. And what's exciting about this process is that, that converting a somatic stem cell to an iPS cell results in an overall epigenetic and metabolic reprogramming of the cells to a more youthful embryonic state. And it also includes features of regeneration or rejuvenation, such as telomere restoration. And so there's been a lot of interest in using the power of these rejuvenating factors or reprogramming factors to improve healthy aging. So for example, in the mouse during aging, there's an accumulation of the aging hallmarks that I mentioned earlier that are well known to occur during the aging process. And delivery of certain levels of these reprogramming factors has been shown to actually epigenetically reprogram old cells into younger cells. And this actually works, at least to some extent, across different tissues and cell types, including stem cells such as hematopoietic stem cells. So another intervention that has been perhaps much more widely studied and is easier to implement is dietary interventions that have been shown to improve healthy aging of stem cells. So for example, caloric restriction, which is a well-conserved sort of rejuvenating intervention, has been shown to also enhance stem cell function with age. And so that's been shown, for example, in the muscle, where calorie restriction or reducing the diet and overall caloric intake in mice has been shown to increase satellite cell number and enhance muscle stem cell function. So, so as a consequence, in these animals that have been caloric restricted, there's an increase in muscle regeneration and transplant efficiency. And so the benefits of caloric restriction are not restricted to just the muscle compartment. It's also been shown that caloric restriction can improve the functionality of hematopoietic stem cells, intestinal stem cells, and can also result in an increase in neurogenesis and cognitive function. Now, caloric restriction is just one dietary intervention that's been studied in this manner. It's also been found that time-restricted feeding or intermittent fasting can improve stem cell function. In animals, ketogenic diets have also been shown to improve stem cell functionality. And as I mentioned earlier, enhancement of certain metabolic cofactors such as NAD through treatment with nicotinamide riboside have also been shown to improve stem cell function and maintenance. And then lastly, rapamycin has been shown to enhance stem cell functionality. And so it's important to note that most of these dietary interventions have really only been tested in mice. And so it's unclear yet exactly how humans will benefit and whether stem cell compartments in humans will actually be improved by these interventions. But interestingly, these interventions have pretty much been shown in every case to function through the mechanisms that I mentioned earlier. So dietary interventions in general downregulate growth factor signaling such as insulin IGF signaling and TOR activity. So so what I mean by this is that they're impacting these nutrient sensing pathways that I mentioned earlier. It's also been shown that these dietary interventions result in an overall metabolic reprogramming that's healthy for the cell, as well as improved immune function to decrease inflammation with age. And interestingly, in recent work, it's been shown that these dietary interventions can also help with the microbiome status. So basically confer a microbiome remodeling during the aging process that is healthy for aging. Okay, so in summary, I've gone over the intrinsic mechanisms of stem cell aging and how they're impacted by a number of extrinsic mechanisms with age. But I think the really exciting take-home message here is that based on what we know from various interventions, there is actually potential for rejuvenation of stem cell pools to enhance healthy aging and improve and increase the number of years that are spent disease-free in humans. And so this is just a list of references to papers that I mentioned along the way for your interest, and thank you very much for watching.